people get confused about what you say and other people think you know what they're talking about, it becomes a problem. So, so even though I sent this to you in advance, you might not really know what the point of it was, but we really do have to think in terms of our proteins as interacting with each other. And the way that nature has created for them to interact is very much like you playing with little Legos. They come with different little shapes that can then bond to each other so that you can get your Legos built together so that you can have a constructed piece that interacts with each other. And so if you just think of these little domains as little Legos, then it becomes obvious that some things can interact with some proteins can interact with other proteins. And the reason we want this is that, first of all, it puts the proteins in organizational units so that those that belong together can work together. And then you need the appropriate timing so that when one protein may get activated, the other proteins then follow suit. And they do it in response to maybe one single signal that can make everything else fall into place. And then the placement. This is one way that you can have differentiated cells that can respond to a series of proteins that others don't respond to. So this is particularly important in signal transduction, where we need to have identified complexes all working together instantaneously in response to a hormone signal. So, so we're going to look at this particularly with respect to some of the things that we're going to talk about today. And I think this really starts with what you had done with your G protein coupled receptors, because the way that our receptors have desensitized is that they responded to G protein receptor kinases that would phosphorylate different parts of the receptor that would then allow beta-arrestins to bind. And we know most of what we've seen has been from a prototype from arrestin from the visual system, but we've got two arrestins that work through the G protein coupled receptors, and they call them beta, largely because the lab, Lefkowitz lab, that really understood this the first time was looking at beta adrenergic receptors. So he just named these beta arrestin, and there were two of them, and so now they're named 1 and 2. So the way these work is that our adrenergic receptors or any other G protein coupled receptors, once the G proteins have dissociated and gone to their effector molecules, that leaves this interior portion free to now complex. And the way this complex is that we get a GRK molecule that can phosphorylate here, and there are 7 of them altogether. 2 of them are only going to be found in the retinal system, so that they only respond with rhodopsin. The others, which are listed 2, 3, 4, 5, and 6, are all going to interact with hormone receptors inside the cells that are the rest of the body. So here we have one that's GERK2. GERK2 combines into this complex by binding to the beta gamma. And in a few minutes I'll show you the plextrin homology domain that it can use to interact with this beta gamma subunit. So once it does, now it becomes part of the complex. It phosphorylates, and these little phosphorylated serines and threonines are then the trigger for binding the beta-arrestin. So once the beta-arrestin has bound, it can now interact with other kinds of proteins. So the GERKs can also interact with other proteins, and this is a good example of the GERK2 that we just saw. It has different domains. It's got its own catalytic domain, and that means that's the part that binds the ATP that provides the phosphorylation to the protein. But it's also got, on its N-terminal side, opportunities for binding to the G-beta gamma right here. And you can see it's got opportunities for binding to a lipid raft protein called caveolin, And also, it's got a binding site for calmodulin. So what does that tell you about this particular kinase?
Calcium?
It can respond to calcium, because if it's got a binding site for calmodulin, then it's going to be having this. The calmodulin is the calcium-sensing protein that provides the information to all these other proteins that we've already talked about. So this one has one of those. And it's got a C-terminal domain that has some other parts that interact with these. So once your protein is all folded, just because you've got a linear segment that can respond to calmodulin, doesn't mean that's the be-all and the end-all. The rest of it is over here on this C-terminal domain. So you can see it's got a place for clathrin, where it can bind to the protein that internalizes. So you can see lots and lots of protein interactions just from this particular kinase and its binding. Now this kinase, the GERK5, and again, these were numbered 2, 3, 4, 5, and 6. This one has a site for calmodulin, and the rest of it is over here on the C-terminal, and it's got a site for the caveolin, which means that it can interact with proteins that are in the lipid rafts, but it doesn't have some of these other sites that you see over here. So I think I can pretty much convince you that it's not only going to be phosphorylating, but it's got sites that that are going to be regulatory. So of these regulatory sites, the one in the lipid raft is negative. So when it's in the lipid raft, it's not going to be working very well. It has to be released from the lipid raft. Our calmodulin is over here too. And so here our calcium is inhibitory on this particular site. But we do have sites for phosphorylation from pKa and pKc, and from the kinase we're going to be talking about today called extracellular receptor signaling kinase, or ERK. So it's got opportunities for phosphorylation that can regulate it as well. So what we're going to look at today is the idea that beta-arrestin, once it has bound to these phosphorylated sites that have desensitized the receptor. So by desensitization, what we mean is those G proteins have dissociated anymore. It's no longer responding to its hormone by having a G protein coupled response. G proteins are gone. So it has essentially desensitized, will not respond to hormone anymore through the G proteins. But it does set up a whole other signaling pathway through these beta-arrestins. So you can see that not only can we have endocytosis, because now it's binding to clathrin and AP2, which are our endocytosis proteins, but we can have it binding to other things. So here's an example of SARC. It's a tyrosine non-receptor kinase, and it can bind right here and initiate pathways through signaling through tyrosine kinase. You couldn't do that before when it had G proteins coupled to it, but you can do it now. And here's a June N-terminal kinase. We'll talk about some of these kinases after the mTOR system, but you can see that now we can set up a whole series of pathways that can go through other kinds of signaling now that we've removed and desensitized it to that, the G proteins. So our beta-arrestin can bind, we can bind to kinases, here we can bind to phosphatases, and set up a whole second series of signaling pathways based on the beta-2 arrestin binding, as opposed to that G protein coupled binding that was really signaling within the first 10 minutes or less. So here we've got long-term binding that can go for hours in response to having the beta-arrestin binding to this receptor. So this is our beta-arrestin pathway. This is the beta-arrestin. We know from taking mutations all the way along the pathway and making them into alanine, they call that an alanine scanning mechanism, that we can find out where some of the active sites are. So we know that there's an active site here for binding phosphodiesterase, and we can tell that by doing an experiment in which we've mutated all the way across the protein and identified where the mutations are that might be involved. So we know from that kind of an experiment that we can have arrestin binding to this phosphodiesterase as well. Kind of clever. Now, when you go to the website that is here on the science website, I've got a lot of these peppered throughout my slide sets. You can see that we can go through here. We can get GERK pathways that have gone through the mechanisms whereby we can ultimately see that we can get our pathway to be quite extensive and have a good deal of networking. So ultimately, after we've phosphorylated at our 7 transmembrane receptors, we inactivate our G protein over here, and this leads us no longer to go through those pathways, but now we're binding to beta-arrestin, and beta-arrestin can take us through some other pathways. So all of this is based on the scaffolding through the beta-arrestins. Now the other scaffold that Group B went through quite a bit on our Friday session are the A kinase anchoring proteins. And we know now that this whole series is activating proteins that are going to be forming now complexes that are like interactomes. They are going to be assembled proteins that are going to be now driving new pathways. So just to show you some complexity here, we have lots of different A caps here. And they're named sometimes by what they are with respect to some of the diseases that they had been involved with. This one is Wiskott-Aldrich, so it begins with a W-A. And the rest of them are named often based on what their molecular weights are, because they were identified by some ability to bind to a pKa, hence the first name ACAP. And then they were found on an SDS page gel to come out at a certain molecular weight. So when you see something like ACAP 350, you know that is a very, very large protein. And look at all of the other proteins that can bind to it. So here we've got our ACAP 350. It can bind to pKa, as can all of these. But now you can see it also is binding to pKc. It can bind to a phosphatase 2A that can take things off, the phosphates off the serines and threonines. Here's a protein phosphatase PP1. This can take the phosphates off the serines and threonines. And here's the phosphodiesterase. So it's assembling all of these together, and you can see this particular one can bind to microtubules. MAP2 is on here as well. And this one binds the pKa, but it also is noted, it's microtubule associated protein is what MAP stands for. It's binding to microtubules. So it can hold the microtubules together so that things that signal through protein kinase A can now interact with those. The one that we first studied, the one with a little M in front, stands for muscle. Muscle ACAP was the one that I showed you bound to the phosphodiesterase 4 along with the pKa. So it could detect where cyclic AMP was already activating this pKa and then remove that cyclic AMP with a PDE4. So you can see in this diagram, it's highly placed near the nucleus, which is where the pKa catalytic subunits would then go in and regulate transcription. So it can clean up all the cyclic AMP that would be around here and turn this off. Here's one that's 220 molecular weight, binds pKa, binds the protein phosphatase 1, and now it's binding another kinase that we'll look at the very last day of class. Here's our Wiskott-Aldrich protein again, but now this one is located near a mitochondria, and not only does it have phosphatases and pKa, but it now interacts with some proteins that are involved in apoptosis. And apoptosis originates by processes that take place in the mitochondria. So you can see it's clearly associated with a function here. But down here, this same protein, this same ACAP, is now bound to actin cytoskeleton and is regulating some other functions. So let's take a look here at some of these individually. This one is an ACAP that has a name. It's called GRAVIN. And it can be regulating from the beta-adrenergic receptor. It can actually be bound to the receptor through phosphorylation sites. So once it's bound, its interaction with PKA keeps it right near where the cyclic AMP is going to be made. This is a beta-adrenergic receptor. And it also has a PKC. So how does the PKC regulate? Once it is released from the beta-adrenergic receptor, now this PKC can phosphorylate its own site here, and this can be a feedback mechanism, a feed-forward mechanism by which PKC functions can now regulate the PKA functions. So we have some regulation here that involves both of those kinases that bind to this particular ACAP. Here's our ACAP called LBC, and this one regulates small G proteins. Now, small G proteins are GTPases, just like your alpha subunit, but they're about half the size because the only part of these G proteins that's left does not include that helical domain that was part of your alpha subunit for heterotrimeric G proteins. So we've got a whole family of these. We'll talk about a little bit later. Here we've got Rho. This is one of those small G proteins. It works just like your alpha subunit. It's got a GDP bound to it. When it binds, it releases this GDP, and GTP can bind and it can be active. So this particular ACAP not only binds to PKA, but it can serve as a GTP exchange factor, or a guanine nucleotide exchange factor for Rho. Once the PKA becomes activated, now we have a different function already, because now it can phosphorylate at a serine on the ACAP, and it disrupts its interaction with Rho. So it no longer can serve that function. Here we've got one that binds to another protein kinase called protein kinase D. So we can have a series of events that our PKC phosphorylates here and can regulate that binding. And then when the PKA phosphorylates, it causes the release of the PKD that's already been phosphorylated by the PKC. So I think you're getting the picture that you can have multiple signaling pathways that can now be generated as these kinases not only provide their function to phosphorylate their substrates, but now can be regulating and coordinating with some of the other kinases. Here's our Wiskott-Aldrich. There's a domain on here for binding to other proteins. The one that has the protein that was discovered first was called verprolin, so we call this verprolin homology, and that's what the V stands for. So this assembles complexes, and depending on whether you're working with the mitochondria or whether you're working with the actin cytoskeleton, the role is going to be somewhat different. So I'm going through this kind of quickly, but you're getting the picture now that we can have different kinds of binding. This one is for neurons. It's binding to one of the ESD-like proteins that has PDZ domains on it. So these little PDZ domains have a function that they can interact with this ACAP 79/150. Now when you see a slash after a name and then another name, that means that there were two different kinds of laboratories studying something. One of them named it one way and another one named it another. And the reason you've got two different molecular weights here is that one was found for mouse and the other one was found for human. But it's a homologous protein. Yeah?
Should we focus on each of these, like ACAPs and the examples, or like general gist of
I think right now you want to get your generalizations. Okay? You want to know what it is when I say a PDZ domain. You want to know what it is when I say a plextran homology domain. Okay? Now, if you want to memorize all of this, you can probably do that. But on the other hand, you probably want to think in terms of where it fits into what you already know and why it might be useful. Now, you might be more interested in why it might be useful in developing drugs. So if you begin to think about this, then it begins to make a little more sense in your brain. So on the other hand, you might want to go back and look up Wiskott-Aldrich disease and find out a little bit more about it and how this might be useful for you. So, you know, if you looked up a disease because that's what you're interested in, then you can find out why this protein was discovered in the first place and what some of the problems are once you have a dysfunctional protein. Okay, so now some of these domains. So now we're getting down to the Legos. The Legos have little circles, the Legos have little squares. You put the squares together with other squares, you get a complex. If you tried to put a square together with a circle, it's not going to work. Okay? That's the complex. What we've got first we're going to talk about is PDZ domains. That stands for the 3 proteins that this was first discovered in. But we now know there are hundreds of proteins, but they were named after the first 3. So here we have one, postsynaptic density 95. And I've talked to you about that before as an example. That's a protein that holds together the NMDA receptors and nitric oxide synthase and actually guanylyl cyclase has a binding site on there too because it's got 3 of these PDZ domains. So we can hold 3 proteins together. The idea is that they need to be together so that they can function sequentially. Discs large, zona occludens. Zona occludens, does anybody know what that is? When you've got endothelial cells and they need to be stuck together, and you need to get things that go from one cell to the other, or between the 2 cells, you've got 2 zones that have proteins that hold your endothelial cells together. Your endothelial cells form a nice long brick wall, right? You know what an endothelial cell does functionally? Let's say inside the vasculature.
It allows movement of things through them if they're fenestrated, or
Right.
provides kind of protective, like you said, wall.
Right.
That can be leaky.
So if you want to get things through, you've got a zona occludens. And the proteins that hold that together have PDZ domains. So here's our little Legos. Here's our PSD95, PSD93, SAP102, SAP97. They're all part of the very same family. And these little purple round circles here are the PDZ domains, if you want to look at them in a bigger picture kind of way. Yes?
Sorry to interrupt, but is this still the review that you said would happen at 10, or is this just lecture now?
We're going to talk about receptor tyrosine kinases in a few minutes.
Okay, so that's
Okay.
the lecture for today.
Yes.
So this isn't the lecture for today. Okay.
Well, it is if you want to know what I'm talking about.
Right. No, I
So
just
like this is the continuation of
yeah,
Right.
this is the continuation.
So we've
Okay.
got SH3 domains right here, and they're going to bind to another signal. You can see if we've got domains that we can actually interact here in our PSD system with 3 different kinds of proteins that would all have ligands. So, you know, again, you're thinking in terms of a receptor and a ligand. So a ligand is going to bind to the receptor. So those proteins that can bind to one of these domains is what we call the ligand. So you're doing protein-protein interactions now. What you thought about when you were doing your receptor binding was maybe a little small molecule like a hormone. Now we're talking about protein-protein interactions. So what I'm going to show you in a minute is what those ligands look like on the proteins. Here we've got some turquoise bars. These are guanylyl kinase domains. Actually, they're not functional kinases, but they're domains that can interact with other proteins. So you can begin to get the picture here that virtually all of these proteins have a domain that can bind to other proteins. And the purple ones are the PDZ domains, but some of the others are going to be binding by other mechanisms. And right now you can see down here, the other one we're going to talk about today is phosphorylated tyrosine binding domain. And there's one right here on this protein. So some of these proteins you'll probably recognize right now. Here's our zona occludens that had 3 of these PDZ domains, but it has some other domains as well. So you can see you can fit your Legos together. You can have your circles, you can have your squares, and that way you can interact with one of these and one of these and put them together. And that's exactly what nature is doing with these cell signaling proteins. So as you go through here, you look at some of these. This was one that one of our graduate students talked about when he gave his dissertation in neuroscience last week. He was talking about the cell signaling through calcium that would be involved in getting vesicles close to the plasma membrane so that they could release neurotransmitters. And you can see here we've got 2 of these PDZ domains and 1 of these protein tyrosine binding domains. And later on today we'll talk about protein tyrosine binding. So here's a list of some of these. So again, if you're going to relate this to things that you know and things that you're interested in, take a look at this list and identify 1 or 2 proteins that you think are important to your research or your career or some disease that you know about so that you can really take this personally. Here's serotonin receptors. Some of you might be interested in serotonin receptors. Lots of people are depressed. You may be depressed. If you're going to take an SSRI and you want to know how your serotonin receptors are doing, you might want to pay some attention to this. Proteins that interact with PDZ, PSD95. So here we have serotonin receptors interacting with the PSD95 that had 3 of these PDZ domains. Some other things, let me see what we might look at. Here's one that's involved in the calcium signaling in vesicles. Here's myosin number 6. So here's nNOS. We talked about that last week. Nitric oxide synthase that's neuronal. So find something that you're interested in and take a look at it. So here are some of those that are going to be moving cargo. Here is SAP-102. It's binding initially in coordination with SAP-97. Here's a microtubule. And so it is transporting proteins by binding to those domains. So it's taking glutamate receptors and it's going to take them to where they need to go in a postsynaptic density, which is what this is represented as. So they're initially being used as a cargo. So the interaction here is the glutamate receptor interacting with this protein through its PDZ domains, and this one would have the PDZ ligand on it. And when it gets phosphorylated by CAM kinase, then it drops it off of this protein and allows it to bind over here to PSD95, which also has PDZ domains. So it's really moving it from cargo during the transport all the way to where it belongs, where it can bind to another place. So this is how we get things all settled up. We've got maybe a different function, but remember, homology, we had SAP97 looking a lot like PSD95 in that they had these PDZ domains. So those are our functions. For those of you interested in the synapse, here you can see we've got interactions that hold the presynaptic side, where we're releasing vesicles, to the postsynaptic side. And because we've got one of these coupled to this PSD95 that has these 3 little purple circles, not only can we keep it near the postsynaptic side where this was named, postsynaptic density, but now we can have it interacting with our nNOS. You know what nNOS is, because we talked about it last week. You can have it binding not through the PDZ domain, but through one of these other domains with a protein that's going to interact with other proteins that are going to get it really close to your IP3 receptor in your smooth endoplasmic reticulum. So you see, if you stare at a diagram like this for a while, you can see how the things we've already talked about can now be assembled together and can actually work together. So here we've got our PSD95s are dimerized, not through the PDZ domains, but through some other domains that can hold them here in the plasma membrane. And in the meantime, they can interact with other proteins. Here's one of these ACAPs, the ACAP 79. That's not binding through the PDZ domain system, but it's binding through, we'd have to go back and look to see what those other little circles were. So look down in the bottom of your note page section on this, and it'll tell you what these little circles are. Okay, so essentially we've got strong connections with proteins that need to interact with each other. In this case, the NMDA receptors, and it's the SH3s. I'm sorry. So that we can get coupling of proteins together. Now, what does this look like? This is on the presynaptic side. You can take a look at that. So when you look at this molecularly, the PDZ domain itself is some helices, some beta-pleated sheets, but the ligand is the last 5 amino acid residues on the C-terminal of the protein that binds to the PDZ domain. So this is our PDZ domain. It's all twisted up. It's not anything you need to memorize. It's the holding hand. This is the part of the protein that's the ligand that's going to bind inside. And it's a very, very simple interaction in that you've got, now the amino acids are numbered backwards, 0, -1, -2, -3, -4. But keep in mind you're at the C-terminal. So no matter how long your protein is, we're counting backwards now. So what you can see is lots of interactions with proteins in this PDZ domain. So the most important amino acids are the ones that are at the extreme end terminal, because you can see lots of hydrogen bonding or ionic interactions with the amino acid residues that are numbered here. So this is numbered 1, 2, 3, 4, and twisted around and in a loop, and 11 and 12, and then just before you get to the helix, number 16, the helix has numbers 13 and 14. So the interactions with 0, -2, and -3 are the strongest interactions. So computer programs, or actually if you wanted to do this yourself, you could, can recognize these amino acids. So they come in certain motifs that we can predict that they're going to be ligands for these PDZ domains. And we can give them little scores as to whether they are going to be likely to bind to certain kinds of PDZ domains. So those scores in the computer can be generated this way. There are some scores that will never happen, and there's a big red mark right there. But in general, if you want to look at this right here at the where we've got our amino acid residues here and our PDZ domain amino acids that are doing the active interactions are the ones in turquoise. you can see where our PDZ ligand is interacting with those. So it makes a nice, clever little fit right there, and you can go through these predictions. Now, the other one I told you I was going to tell you about is the phosphotyrosine binding domains. And there are two different kinds. One is called Sark homology 2, and the other one is called the protein tyrosine binding. So in a few minutes, we'll get to the kinases that actually phosphorylate on these tyrosines. So this is how proteins regulate and recognize them. So they regulate and recognize something that has a phosphate on a tyrosine. So the first one is our phosphotyrosine binding domain, and they call these PTB domains. And here you can see some usefulness of this. This is our complex of a tyrosine kinase receptor that we'll talk about in a minute. But you'll see it's got all of these proteins bound around it. And they're called into place because they can interact with the tyrosines that have already been phosphorylated. And then they are the complexes that are going to go on and do the signal transduction. Here's one that is from interacting with a cytoskeleton. Here are some that are involved in endocytosis processing. And they're all going to be working through a phosphorylation and a phosphotyrosine. So again, pick out one of these that you think is important for you to remember and use it as a prototype. So here are some examples of some phosphotyrosine binding domains. So these are going to be binding to proteins that have a phosphotyrosine on them. Here's one that might be of interest to some people who are interested in regulation through G protein receptor coupling. And you can see they've all got a green square for phosphotyrosine binding. Okay, so how does this one work? So this one has our ability to interact with its domain, and this domain is a helix and a beta-pleated sheet. And the ligand, again, is going to be, here we've got a C-terminal, and we've got some residues. And here is our tyrosine. And it's going to be recognizing this tyrosine that has the phosphate group on it, some amino acid, it doesn't matter which, a proline and an arginine. And this is all going to be assembled so that these can interact with the amino acids that are part of this domain. So here you can see this is our helix. Here we've got our, where's our beta 5 on here? Turning around so that this is our ligand that can interact with this purple helix down here and this sheet right here. So it wedges right in between here and has not only the phosphotyrosine, but the other signal that it's going to be binding here is the proline and the arginine. Okay, so SARC2 are simply looking at plain old phosphotyrosines, and I don't have an example of that, but their SH2 domains are always also going to be looking at the phosphorylated tyrosines. So now I've used the word plextrin homology. Again, this is based on the first protein that was discovered to have this kind of binding mechanism as a domain, and that was plextrin, which is a protein found in red blood cells. And by homology, that means that all of these other proteins that we now have, we're going to call them pH domains, plextrin homology domains. And this one, you know, don't memorize the structure. I'm just giving you something that you can hang your hat on. These bind lipids, or they bind very hydrophobic regions of proteins. Could bind either way. So here we've got what we talked about last week was our phosphatidylinositol that has multiple phosphates on it. And remember, I told you those were signals for binding cytosolic proteins to get them to come to the membrane. So here we have our phospholipid. Here's our phosphate head group on our inositol. Here's our two acyl chains in the membrane. And as we get phosphorylation on either the 3 or the 4 or the 5, or in this case there's a 4 and a 5, or it could be a 3 and a 4, or it could be all 3, 4, and 5. Like one of those multiple choice questions that Dr. Petrovic gave you, where you had multiple choices. Well, the proteins have multiple choices too. And they can tell how much phosphate charge is here, and it's all based on charge distribution. So you can see why the proteins might come to the membrane based on binding to lipids, or maybe they could recognize a very hydrophobic domain on a protein. So here we've got some protein examples. I've given you protein examples for almost everything so that you could find something you're interested in. Here's a phospholipase C gamma. So we already talked about that. That's going to be important to come to the membrane because it's going to be the phospholipase that breaks down IP3 and diacylglycerol for signal transduction. So that's a very important reason to call the phospholipase gamma from the cytosol where it exists over to the membrane where it needs to work and where its substrates are. So we have a situation where we need to recognize this phosphate, this positive charge, negative charge interaction, because what we're actually doing is looking for the interactions with the IP3. So here we have an inositol. It's got its head group at 1. It's got 3, 4, and 5. It's coming out of the membrane, and it's binding to these loops that are part of this plextrin homology domain. So here's the loop part that's regulating and recognizing the 3-hydroxyl phosphate, the 4-hydroxylated phosphate, and the 5. And when you look at how this is interacting, it's got a very large positive charge density. What kind of a charge does a phosphate have? What?
Negative.
Negative. So what you're looking at is this positive charge density on this protein that's going to be highly attracted to the 3 negative charges that you have on this inositol phosphate in the membrane lipids. So it's going to come based on its little group up here that's going to be positive. So here we've got some of these proteins that we just talked about. Here's the phospholipase that we talked about here. It's going to be having a positive charge domain. All of these do, but they're a little bit different in shape. So the strength of the binding and the shape of the area that can interact really tells you whether it's going to recognize the 3-4, the 3-5, or the 4-5, or the 3-4-5, and what kind of binding it's going to be. So we get this binding to our phospholipids. Here's examples that you can look at of 3, 4, 5. This one may not be binding to the 5 position, but is going to be very keen on interacting with the 3 and the 4 position with those amino acids there. Here's a phospholipase that's interacting with 5 and with 4, but not with 3. So here we've got one that is nestled in here. You've got the 1-phosphate, 3, 4, 5. These are all calling it to the membrane where it can interact with this lipid. Here we've got a protein with a plextrin homology and a PTB domain. And so you can see it's got a region where it's binding over here to some proteins. And then the inositol 1,4,5 phosphate is calling it to the membrane over on this side. So that's 2 different domains. working two different ways. Sometimes your domains are going to be separated. Here we've got part of the plextron homology domain on the N-terminal. We've got a PDZ domain here in the middle. Plextron homology, the rest of the domain is over here. So when the protein is all folded together, it's got what would be the complete plextron homology domain, but if you looked at it sequentially, you wouldn't be able to say, aha, this whole thing represents that domain. Okay, so I've introduced you to some of these domains. When you go back and study your NNOS, and you remember that I told you it combined to the PDZ domain on PSD95, so it can be really close to the glutamate receptors. That means something to you now, right? Maybe it didn't mean something last week. Sea of dead faces here. Nod your head. Endothelial NOS. Remember I told you, you go back and look at it, it doesn't have a place to bind to a PDZ domain, but it binds to plextron homology domains. What does that mean?
Will eventually have simulation
It means that it was cytosolic, and when it got ready to bind to the membrane, it was in response to these plextron homology domain lipids that we just talked about. So the lipids get phosphorylated, and now the NNOS can come to the plasma membrane.